This is a production of Cornell University.
I will be presenting on the work I've done for the last 3 growing seasons here at Cornell, working with Justine Van Heuvel, with Thomas Bjorkman and Anna Catherine Mansfield, also on my committee. The work here for my master's has centered around using undervine cover crops as an herbicide replacement in cool climate Riesling vineyards. So I'll go quickly over the background. Why am I even bothering to look at this? For my materials and methods, I did all my experiments at 2 field sites, so I'll go over the design at both of them, my different results, and at the end of the day, what can we say about this work? So currently, it is pretty commonplace to use cover crops at some point in the season between vine rows, but it is almost universally kept weed-free or vegetation-free in the zone directly beneath vines. This bare soil strip is maintained with a combination or selection of herbicide and or cultivation. This is done because growers perceive that it would be a very negative effect on vines having vegetation there. It's not so much supported in the science literature, but it's definitely the perception, and that is why it's done. So any risk, there's always a risk when you introduce an herbicide to a system. Not only are you increasing your chances of resistance in the weed community, but you're always offering the opportunity for these herbicides to leach out or run off. This is of special concern when you're growing vineyards above lakes, like in this region, and if you're worried about contaminating your groundwater sources. And regardless of whether you're using herbicide or cultivation, bare soil is a big risk in terms of your soil health. Any kind of bare exposed soil is going to be subject to crusting during a hard rainfall event, which leads to further runoff. You get soil structure degradation, a decrease in organic matter, and also it's very subject to erosion anytime soil is left bare. This is of great concern when you have vineyards planted on hillsides, which is very common not only here in the Finger Lakes, but worldwide. So this is all coupled with the fact that if you look at kind of the state of the union of vineyard growth habits in this region, we have a lot of very excessively vegetative growing vines, for lack of a better phrase. You can see that picture there. The shoots have not only well extended beyond the trellising, but they're starting to grow into the next row. We have frequent rainfall throughout the growing season here, so water is rarely a limiting factor. Many vineyards are planted on what just happen to be very deep and fertile soil sites, so that's not a limiting factor. And we also just have selected, due to commercial and production reasons, some varieties that just have a particularly vigorous growth habit. So all of this sort of creates the perfect trifecta of growing more like jungles than little vineyards. This excessive vegetative growth is a problem because it is associated with very poor resulting grape and wine quality. Shaded buds are less fruitful. A lot of the desirable aromas we want in our wine are correlated with sunlight exposure. So when you have 15 bazillion leaf layers blocking them, you're not going to get a lot of exposure. And those conditions are really ripe for disease. All that leaf area is going to contribute to a higher relative humidity and just make it harder to get into the canopy to manage your fruit. And so given that deleterious effect on the vines from this excessive vegetative growth, we spend a lot of time and money in vineyards maintaining it. We'll go through in pretty much right from the time shoots burst and have to remove vegetative growth. We'll take out shoots, we'll leaf thin, we'll drop excess clusters, we'll drive tractors throughout the entire season to hedge off those tops of shoots. But this is all costly and takes lots of time to manage. And it's also, at the end of the day, just treating the problem, not the source of the problem. So there's been successful research in other humid climates that have this excessive growth problem in New Zealand, Long Island, and Virginia that have shown that when you replace using herbicides directly under vines and instead plant a very intentionally planted undervine cover crop, not only are you eliminating herbicide use, but you're actually getting a positive devigorating effect. However, as this Californian can tell you, the Finger Lakes is pretty cold, and we aren't as warm as these other regions. So we have sort of a different management practicality to work around. In the winter, we mound up a lot of soil around the graft unions of vines. The European grapevines you guys are going to be familiar with, including Riesling, which is what my research is done on, are all planted on rootstock. So if we ever had a really hard freeze, these vines would kill to the ground, and all you're going to be left with is your rootstock, which isn't going to produce your fruit, and you're pretty much back to square one. So by hilling up this dirt all over those graft unions, you're ensuring the survival of some scion buds, which Come the spring, you hill back down, you can grow up your Riesling or your Cab Franc or whatever it may be from those surviving buds. And you're not back to square one. You still have some work ahead of you, but you're not devastated. Also, at the end of the day, crop insurance requires growers to hill up. So regardless of whether you're going to take the chance or not, you're pretty much going to do it. So given this need to hill up vines in the winter and hill them back down in the spring, we have to operate on kind of a different system, vineyard floor management system, than these other regions, which showed success with looking at permanently established perennials. That's why my objective for my master's work was to look at annually established undervine cover crop species as an alternative to herbicide in Finger Lakes Riesling Vineyards. We hypothesized that given this very common excessive vegetative growth habit of Riesling grapevines in the region, that there will be no deleterious effect by introducing this undervine cover crop vegetation with the vines. We actually, if anything, predict that there would be a positive outcome by reducing this vigor. So my data sort of stemmed on two sides of looking at the question. The first was looking at different measures of vegetative growth right now, and subsequently taking some rough measures of vine nutrient and water status. For water, I looked at Midday and PreDawn stem water potentials. I'll talk about those later. Take a quick moment, because I'll mention it several times. Enhanced point quadrant analysis, or EPQA, is a way of quantifying the canopy characteristics, the number of leaves, the number of clusters, those sorts of things, and the light environment within the canopy. That's what Jim Myers did for his PhD, a former student of Justine's. He's now a postdoc for us. So that's building upon his work. And then petioles were collected at Verizon for nutrient analysis for that side of things. Then I don't just watch the vines grow all season, but I do in fact harvest the fruit, and different measures at harvest are taken, and then the subsequent juice is analyzed. Brix is our measures of sugar, so the soluble solids in the juice. Sugar is really important because in fermentation, sugar goes out to alcohol. TA is short for titratable acidity. This is really important for your perception of acid in the wine and juice. pH, not only is it related to acid, but really important for microbial stability in wines, so you don't have bugs that we don't want growing in there and tainting your wine. And YAN is short for yeast assimilable nitrogen. This is important because yeast are biota, a living organism, and we want them to do our bidding of converting sugar to alcohol. So we need to provide them with enough nutrients for them to do that. When you have low nitrogen values, fermentations can get stuck and sluggish, putting off aromas, and at the end of the day, not completing your fermentation. So that's why it's an important value to look at. And I also subjected these wines to a sensory analysis, looking at differences perceived in wine aroma, which I'll also talk about later. So to give you an idea of the abbreviated season we're working with, in May we get bud break. This is when grapevines are dehilled. That soil is pushed back down from the roots or from over the graft union. This is also when I begin to establish my undervine cover crops. By bloom, which occurs in mid-June, you can see that's the second picture at the top there. We're not pretty like the apples, but it happens nonetheless. The cover crops have emerged. This is also typically when growers will start having to manage any kind of excess vegetative growth. They'll start going in and shoot thinning. They might adjust cluster crop, that kind of thing. This is also when I start looking at the water potential readings, and I start tracking shoot lengths. Veraison is a really critical time in our world. Veraison is the fancy word we have for when ripening really starts to accelerate. This is when sugar starts to rapidly accumulate in the fruit, acid levels start to drop. In red varieties, you get the green to red color shift change. And in the whites, you do get a color shift. It goes from those bright, like hard green berries to the skin soften, and in Riesling it turns a nice kind of bright yellow color. Harvest roughly happens late September to most likely early October. This is when all the fruit's taken off the vines. I take different yield components. I crush the fruit, analyze the juice, and the juice is then put through fermentation to be completed wines. By November, we are hilling our grapevines back up. And this is sort of would be the end date then for my undervine cover crops, because we have a giant cultivation operation underneath the vines. At this point, the vines have pretty much gone dormant and have leaf drop, and they're preparing to hibernate in the winter like I wish I was. As I said, I do make wine from the juice. This is sort of a standard white wine fermentation procedure that in general was held for both of my lots. What's important to note here is that for each treatment, there are duplicate lots of wine made. And the whole idea is, since this is a viticultural trial, I'm trying to be as hands-off as possible in the enological process and try to allow characteristics in the vineyard to be carried through. So I'm using a yeast that doesn't throw off a lot of interesting aromas. I chaptalize or add sugar to a standard sugar level. I supplement the yeast or the YAN level to make sure these yeast are going to be able to go, and I do ferment these wines to dryness. So for the multidimensional sorting analysis by wine aroma, the big fancy word for my sensory tests, in short, all the duplicate lots were presented to panelists, and they were asked to sort wines by similarity into groups. If you found a wine to be similar to another wine, you put them in the same group. If you found them to be different, you put them in different groups. This gets a lot of grouping data that's then run through SAS and can give you a visual representation of the differences, which I'll be showing you later. So I had 2 field sites that I did my research on. The first was a commercial vineyard on the east side of Seneca Lake. These vines were older, almost 20 years old now. I planted on a honey silt loam, Riesling. And what I want to note here is it's a Scott Henry training system, meaning that there is both an upper and lower fruiting zone. This is important because we'll often do this in vineyards that have sort of excessive growth, but this already has been done at this vineyard and we were still seeing excessive growth. There were treatments. The undervine cover crops were originally planted in 2011 and carried through this year in a randomized complete block design with 4 replicates per treatment. My 4 treatments were as such: annual ryegrass planted in the middle of May. This is a later establishing. While planted in the middle of May, it doesn't really get going, and I noticed the most biomass accumulation much later in the season, such as July, August, and it grows well through October. Buckwheat, that beautiful white flowering plant, seeded in late May. Interesting about buckwheat is it frost kills, so that'd be ideal for a kind of abbreviated time frame that we're looking for. Buckwheat also has a lot of notoriety right now because work out of Berkeley and work by Greg Loeb here has shown that it could potentially increase the incidence of generalist predators in the vineyard and increase rates of parasitism of vineyard pests. There's a lot of interest around buckwheat. Natural vegetation, polite word for weeds. We allow them to grow throughout the season, whatever may be there. And then our control is the traditional glyphosate spray applied 1 to 2 times a year. The undervine cover crop treatments were mowed as needed to keep them from getting too tall into the canopy, and that ended up being 1 to 2 times a year. My second site is the Cornell orchards at Lansing, in one of the Riesling blocks that I kind of inherited from Ian Merwin's old work. Here, these vines were originally planted in 2007. It, once again, is a 2-tier flat bow, which is a double canopy fruiting system. And that picture that I used on the slide about excessive vigor came from this site, so I can assure you that there's a lot of excess vigor here. These treatments were originally established in 2012 and carried out this year. This site's a little complicated, so I'll do my best to describe it. Undervine cover crops were planted down the entire lengths of rows. Each cover crop was planted down 4 rows in its entirety. There is then an overlay of irrigation at this site that allows me to irrigate half of a given row. Which half is irrigated alternates down the rows. The whole site is in total just under 1 acre, to put that size into perspective. I initiated irrigation when the average midday leaf stem water potential exceeded -8 bars. This is a rough threshold for when vine water status tends to be inhibiting vegetative growth. So that's kind of where that number came from. In 2012, we irrigated 3 times. In 2013, it never actually got that dry to need to irrigate. So I did analyze it in case there was any kind of a carryover effect with the irrigation, but that was just simply the case this year. My 3 undervine cover treatments here was the buckwheat, which I talked about, the same glyphosate sprayed control. But what I did here was look at chicory, which is sort of a different growth habit. All the rest are pretty much straight annuals. Chicory is a biannual, which often acts as a short-lived perennial. Its first year, you can see it had that nice low-growing rosette form, established really nice. This year, even though I reseeded and there was a complete cultivation operation, obviously some survived, and it shot up all these, not only the low-growing rosette form, but all those flowering stalks. Another thing to note about the chicory that makes it sort of different is all the rest tend to be relatively shallow-rooted cover crops. Chicory has this nice deep taproot, and these were taken very early in the season. When I was doing soil sampling just a few weeks ago, I found chicory roots going past even 20 centimeters in the soil. So it's just looking at a very different growing habit of a plant. So first question, were my cover crops there? Why look at this question if they weren't? So yes, in short, at all 3 sites, the cover crops established. 2011 at my first site, I had mowed once already, so that's why they have much lower values. I think that's of interest to note. In case a grower was ever worried about it going out of control, mowing does sufficiently decrease a lot of the biomass present. In both other years, you can see annual ryegrass and buckwheat established well, but with a fair amount of weeds. And that in all years at both sites, the control was essentially vegetation-free. You can see the biennial growth habit of chicory reflected in the biomass, with it being relatively low in that first year, but a lot of it was present this last year, and buckwheat was relatively consistent. So one of my first measures of vegetative growth was looking at cane diameter. This is a bit of a Goldilocks story. When we go to prune in the winter, we don't want a cane that's too thick or too thin, and the rough ideal that some people argue is about pencil thickness, or 6 millimeters. At my first site, there were never any significant differences made on cane diameter. But at my second site, in this last year, chicory actually did significantly reduce the cane diameter down to 6 millimeters from just around 7 compared to the glyphosate control. So it might not be a big difference, but it definitely was a lower value that's closer to what we would consider more ideal. As I said, I tracked shoot lengths and could subsequently calculate shoot growth rates throughout the season. For my first site, there was never an effect in 2012, but this last year, natural vegetation actually did have a greater shoot growth rate in the early July time period. And at my second site, there was never any significant effect on shoot growth rate by the undervine cover crop treatments. So this is this EPQA I was talking about earlier. At the first site, the annual ryegrass, the buckwheat, and the natural vegetation never affected any of the canopy characteristics. Same goes for in the first year at site number 2. However, in this last year, chicory did significantly affect canopy characteristics. It reduced the total number of leaf layers in the canopy. It reduced the occlusion layer number, which means the number of shade-producing layers in the canopy. And then, not surprisingly, if you have fewer shade-producing layers and fewer leaves, it actually increased the cluster exposure flux availability, which is a big phrase for the light exposure on clusters. This graph here, as you move left to right on the x-axis, you're moving from deeply shaded fruit to highly exposed fruit. So in our world, we tend to want more exposed fruit for disease management purposes. You can see that the tall blue bar and the yellow bars are the control treatments, and they by far and large were dominated by deeply shaded fruit. Whereas when you come and look over here at the more exposed regions, it was the 2 chicory treatments that had much more exposed fruit, and the buckwheat was yet again an intermediate. When we prune in the winter, we'll calculate all that growth from the last season and weigh. In the first site, there was no significant difference among pruning treatments in 2011 and 2012. 2013 data will be coming, but I don't have that for you yet. And then at my second site, what's interesting here is that there was actually a significant cover crop by irrigation interaction effect, where the irrigated control yielded a significantly higher pruning weight than the non-irrigated buckwheat. But that's the only definitive relation that really came out in that. So PreDawn water potential, which at this point I think almost everyone in this room has heard me complain about, is when I go out to the vineyard at 3:30 in the morning because there is no transpiration on the plants. And this is when the vine water status will have equilibrated with soil moisture. And so I'm getting a rough measure on soil moisture where the rooting zone grape roots are. In 2012, there were no differences. In 2013, there was one significant difference at my first site. It actually did happen. The control was significantly, slightly less water stressed. The more negative the value, the more water stress. But I would just like to point that negative 1 1/4 bars to negative 1/2 bar is hardly what we would call water stressed. As I said, -8 was the limit that we began to see an inhibition of vegetative growth. So these are just essentially very well hydrated vines. Same goes for site number 2. There were never any significant differences in this PreDawn water potential. So going from the least amount of stress to the most amount of stress a vine is seeing, midday stem water potentials are taken at solar noon and kind of looking at the maximum potential water stress. For site number 1, there was never any significant differences in the midday stem water potential among the cover crop treatments. For site number 2, there was never any significant difference in the midday stem water potentials among treatments. Those black arrows indicate when irrigation was initiated in 2012. And as you can see by the graph, 2013 was just a much wetter year. We just never received that level of water stress. Those vines were very well hydrated, even with the presence of an undervine cover crop. So to look at, see if there's a potential nutrient effect of having these undervine cover crops right there in the rooting zone of the vine, we took petiole tissue at Veraison, sampled and submitted for analysis. 2013 data is still pending, but this is the data from 2012. The overall story here is that there is no overall story. There was no consistent effect that I could find. Interestingly, annual ryegrass actually increased the levels of phosphorus compared to the herbicide control and increased levels of iron. At the second site, there was no effect on any of the nutrients except for nitrogen. And you can see yet again there was this interaction effect where the irrigated control had significantly more nitrogen than both the buckwheat treatments. So at harvest, we look at different measures of yield. Look at the total number of clusters per vine, so a lot of counting. And then we weigh these total clusters on a per vine basis to get a yield per vine. You can then calculate an average weight per individual cluster. And then from those clusters, I take individual berries and weigh them out so I can get an average weight per berry. And then from that, you can calculate on average how many berries there were per cluster. So for my site number 1, there was never a terribly consistent effect except for that annual ryegrass did influence the number of berries per cluster, having significantly fewer. Same with the cluster weight was slightly smaller, but significantly so than actually the buckwheat treatment, but no terribly consistent effect. I'll take a moment here to explain what the Rivaz index is too. That's a very common measure in the grape and wine community to help evaluate the ratio of reproductive to vegetative growth. What we do is on a per vine basis, take the total yield in fruit, like how much that fruit weighed in kilograms, divided by the pruning weight we harvest that subsequent winter from the vine, and kilograms. So it essentially ends up as kilograms over kilograms or a unitless ratio. And what exactly the ideal ratio is, that's a wonderfully debated subject. Roughly 10, some people say anywhere between 4 to 10, some people say 10 to 12. But you can see here there was no significant effect on this ratio at this site.
Lindsay, so that's ARG's annual ryegrass?
Correct. Annual ryegrass, buckwheat, control for the glyphosate, and nat short for natural vegetation.
So you're seeing it drop?
So yeah, I did see fewer berries per cluster, and in 1 year I saw a reduced cluster weight. For site number 2, in 2012, there were no differences in any of these components, but in 23, Chicory did have a pretty consistent effect. So while the total number of clusters per vine was not affected, the yield per vine was significantly reduced with Chicory present under the vines. So if you do that math, it's not too surprising to figure out that the cluster weight, the individual clusters weighed less when the Chicory was present. Subsequently, also the berries were smaller for the Chicory treatment, and the total number of berries per cluster was smaller. And I will get back to you on if there was any difference in 2012 for the Rivaz index, but there wasn't for 2012. So this fruit is then crushed into juice and analyzed. For site number 1, the annual ryegrass, buckwheat, and natural vegetation compared to the glyphosate spray control had no significant differences on the Brix, so the sugar content, or titratable acidity, the pH, or the yeast assimilable nitrogen of the juice. For site number 2, for 2012, what was interesting is at the beginning of the season, the buckwheat actually had a significantly lower acid level, lower titratable acidity than the glyphosate control. But by the end of the season at harvest, it was actually the Chicory that had a significantly reduced titratable acidity by just under 1 gram per liter. There was never any cover crop effect on the sugar, the pH, or the YAN. So the wine aroma sensory, as I said, once you do all this grouping data, you get a visual representation of how people found the wines to be similar by aroma. So the closer 2 points are together on these graphs indicate the more similar by aroma they were found to be. In 2012, you can see that while buckwheat and natural vegetation were found to yield more similarly aromatic wines, they were all the undervine treatments were found to be different by aroma than the control. And in 2012, that same trend kind of continued with the buckwheat and natural being found to be pretty damn different than the control by aroma. So for my second site, here I'll note that actually at the previous site, there were 8 wines presented to panelists. Like I said, duplicate lots of wines. There were 4 treatments, so 8 wines were presented. For this site, there were 12 wines presented because I have 6 treatments: buckwheat irrigated and non-irrigated, Chicory irrigated and non-irrigated, and my control irrigated and non-irrigated. 53 panelists participated, so thank you. And then you were asked to sort them between 1 and 6 groups. I don't know if I could have created a more equidistant point graph than what the numbers gave me, but that was what happened. There's potentially a trend here with the control and buckwheat non-irrigated at the bottom being kind of similar in a group Typically, you want to analyze these graphs by groupings, but it's a little rough here. And similarly, the control irrigated and the chicory irrigated potentially. But overall, what is interesting to note is that these wines were found to be different aromatically, and that the irrigation and cover crops were the things that made them different. So at the end of the day, what can I say? It's important to note that these undervine cover crops did yield significantly different amounts of above ground biomass. This is important if you're looking to adopt this as part of your vineyard floor management system, understanding that these cover crops are establishing different depending on species. Overall, undervine cover crops did not significantly impact measures of vine vegetative growth, the two exceptions being that in this last year, natural vegetation essentially doubled the shoot growth rate at one point in the season, and that chicory, in its second year of establishment, reduced cane diameter and the number of leaf layers in the canopy, which resulted in greater cluster exposure to light. Undervine cover crops were not found to significantly affect PreDawn or Midday stem water potentials. And similarly, there was no consistent effect on the petiole nutrient content taken at veraison. These points are important to note because the whole premise of why growers are spending a lot of money and a lot of time to keep a vegetation-free zone under vines is they fear there will be some kind of water or nutrient effect on the vines, which my work has at least preliminarily shown and confirmed with past work that this just isn't the case, at least in this region. Undervine cover crops, on the whole, were not found to also impact harvest yield. This is important if a grower, especially a lot of growers here, are not making tons of money. Their costs of management are quite expensive for any of the viniferas, including Riesling. So they cannot take a hit in yield potentially. So this is important to note that for most of the species tested, there were no impacts. The one major exception being that chicory in its second year of establishment significantly reduced vine yield and cluster size by 25%, and that the berries per cluster and berry mass was reduced by 14%. I will interestingly note, though, that I am not in the viticulture is not the business of optimizing yield. We're in the business of optimizing whatever the winemaker perceives as the wine quality. And there is often a notion that too much yield will have detriment to wine quality. So currently, many vineyard managers pay lots of money to have crews go through and drop crop. So this reduced yield actually may not be quite the deal breaker most people are thinking of in our industry. That could actually be saving you a pass in the vineyard with your crew. Similarly, some people do prefer smaller berry size, so this reduced berry mass could be beneficial if that's what you were looking to achieve in your vineyard. Overall, undervine cover crops were not impacting your juice composition, with the exception being that chicory at harvest did reduce titratable acidity by 1 gram per liter. If you're happy with your juice characteristics, then hey, it looks like undervine cover crops probably won't mess with that, except for maybe chicory. Here too, I would argue that a 1 gram per liter reduction could be beneficial. I don't know if anyone here has tasted our wonderful Finger Lakes Riesling, but sometimes they can be a little acidic. Dropping this by 1 gram per liter could be desirable to certain winemakers' goals. For a few of my data measures, including the pruning weights and petiole nitrogen, there was a significant cover crop by irrigation interaction. This is sort of interesting to note because it's indicating that these undervine cover crops could have varying effect in different soil moisture conditions. So understanding your specific site and any variability in your site could be really critical if you're looking to adapt this to your vineyard floor management plans. And the one consistent effect that we saw year after year was that wine aroma was impacted by the presence of undervine cover crops in all years. Why? Since I just talked about a lot of how my data was not significantly impacting measures of vine growth or water status or nutrient content. I have a few theories on this, so bear with me. That EPQA measurement I took that evaluated the canopy characteristic and light penetration of the canopy, I took once at veraison. Similarly, I took nutrient status of the vine via petiole tissue sampling once at veraison. It is known from work out of our lab, Misha, who just graduated with a PhD from Gavin Sachs in food science, he did work with Justine that showed the timing of cluster light exposure can dramatically affect concentrations of TDN. TDN is that very characteristic petrol gasoline aroma that Riesling is sort of famous for. So that not only did the timing of cluster light exposure impact these important aroma precursors in Riesling, but the overall cluster light exposure, Jim Meyer Myers from our lab just put out a paper showing that very important aroma precursors in the fruit that create a lot of characteristic Riesling aromas are impacted by the quantity of cluster light exposure. So I only looked at this potentially different, these differences in cluster light exposure at one point in the season, at veraison. If there were differences earlier in the season or later in the season, they could have had dramatic effects on aroma precursors, and I was simply unable to detect them. Same idea with nutritional differences. If there were a nutritional difference early in the season or much later in the season than veraison that was affecting the biosynthesis of these aroma precursors in the grapes, I wouldn't have been able to detect it. So those are sort of our biggest theories, especially when it comes to the cluster light exposure. But another thing not to rule out is that green volatiles could be impacting wine aroma. I am introducing a lot of vegetation directly underneath the vine. Some very preliminary work from Justine and Dr. Gavin Sachs has shown that hedging materials directly under the vines can impact certain characteristics of wine. So kind of taking it the next step further too, we also know that decomposing plant tissues do give off green volatiles. And like I said, I've introduced a lot of it into the vineyard. So there is that potential interaction going on. So where do I go from here? I've spent 3 growing seasons studying this. Well, I've actually just received a Northeast SARE graduate student grant to continue my work at my second field site, the site at Lansing, looking at if there's a potential more long-term 4+ years of effect of undervine cover crop establishment. So I'm excited to be able to continue that work there. Similarly, since we do know that these wine aromas were perceived as very different when undervine cover crops were present, doing the more chemical side of things, looking at potential compounds that could be responsible for these differences. Further preference testing. So all I can tell you from those graphs is that the wines were different. I can't tell you if that was a good or bad thing. So preference testing would allow me to better analyze that and understand potential consumer reactions of being sold wine that was grown without herbicide. Similarly, we're still sort of on the search for the holy grail of cover crops. Chicory is showing some promise, but an undervine cover crop that could compete on a level to actually see this reduction in vegetative growth would still continue to be a goal, since we do still have this excessively vegetative growth habit in the Finger Lakes. And lastly, understanding how your vineyard floor management decisions, including using herbicide compared to an undervine cover crop, are affecting the biota of your entire agroecosystem could be something that would be very interesting. These are works cited. We all in science stand on the shoulders of the giants before us. At this time, there's so many people I'd like to thank. I especially want to thank Eric and the Cornell Orchards crew and John Wagner out at Wagner Vineyards for allowing a graduate student to run amok in their vineyards and making their life, I know, more difficult. But without their hard work, I wouldn't have been able to do any of this work. I want to especially take a moment to thank the Cornell Dreer Committee, because I am the fortunate recipient of the Frederick Dreer Award this year, and I'm basically counting the days till I will be leaving for Marlborough, where I will be going off to Sauvignon Blanc country for quite a while, where of all things I'll be doing some undervine cover crop research. And I've also accepted a lab tech position with Indivin Wine Company to work the 2014 harvest over there. But never fear, I'll be back. I've accepted continuing on for my PhD, working here with Justine to continue on with some of the work that I've done here and presented today to you for my master's. At this time, I'd be happy to take any questions, and thank you everyone for listening. Definitely. So the question was in relation to the fact that the annual ryegrass treatment exhibited higher levels of phosphorus and iron, and had certain yield effects.
So you didn't bring it up at all in your conclusion?
Well, yes, it was not a predominant effect. The number of berries per cluster was significant, but not on a level that would I would have been shouting from the rooftops. And it wasn't consistently. It was only the 1 year that we saw a reduction in cluster size. So my sort of response is that yes, we certainly think the soil microbiota and all that could be dramatically affecting it. The soil side of the undervine cover crops is more the project of, it was Becky Siriani and Adam Carl in our lab has taken over and looking a lot at that, and that's sort of it. But definitely it's something to consider. I think what's unique in this situation that could be dramatically affecting the soil biota is the fact that we have hilling up. So we're essentially doing a giant mulching of these decomposing or cover crops into the soil every winter. And then we're mixing it all up again in the spring. So I think that's potentially where you're getting some of these interesting effects, and how these plants are decomposing too. It could have some dramatic effects. So certainly it's something of interest. And yeah, those results were a little surprising, and I'm still waiting to get results back from our Cornell nutrient lab, and when I get those, I'll be able to further see if this is a consistent year-to-year effect. The questions about training of the sensory individuals, in short, they weren't. So for this sort of study, you do not want trained individuals. They are given an explicit set of instructions. They are allowed to ask me questions, and I can expound on the procedure. But the idea is that I am looking for a just normal white wine consumer. The one limitation was that on average you drink white wine about once a month. I was looking for wine drinkers, not just anybody. But this wasn't what you might be thinking of, which is called a descriptive analysis panel, where you have explicit training on the different characteristics. You'll have training on what you think the characteristics are in this type of wine, and then subsequently the standards of lemon flavor or standards of acid flavor and those sorts of things. This was sort of not supposed to be that. This is about your own perception of the aroma. I'm not looking to identify aromas, so it's however you perceive them to be different, you group them accordingly. So the question is there a tendency in difference testing to find more differences than may exist? That's always the risk when you're asking someone to find differences. They can be prone to that. This is a little more, I would say, they are presented with the option of putting them all in one group, which people did if they found that there were no differences. So while it is always a problem, there's always a problem like that, I think, with almost any sensory test you do. There's going to be some bias that way. So I think this test was pretty representative of being able to determine differences. But like I said, at the end of the day, all I can say is differences. I can't tell if they were preferred or not, or while relatively they might have been different from each other, but if overall, compared to all the wines in the world, they weren't that different, I can't say that because I didn't have any other externally tested wines. So there's a balance that way. So the questions about mycorrhizal biomass in where I'm having these cover crops, potentially a nutrient effect, I have not looked at that in any kind of scientific form for me to actually say there was a difference or anything. Doing quick research on these cover crops, there is that potential, especially like with chicory. I definitely know just doing my soil samples, I saw them. But and I'm still waiting to get some of the research potentially back this year about the chicory. But in short, I just don't know. That's on the soil side of things, and that's definitely a direction that would be very interesting. What would I recommend to the grower? I would say if you're a grower who is looking to reduce their inputs, which I think every viticulturist should be doing, and if you are relatively happy with your vineyard, I think looking at these annual undervine cover crops, especially something like buckwheat, which was pretty well controlled, the weeds were relatively low, it established very consistently year to year, and I just was not seeing effects, I would say why not try it? Why risk using herbicide over and over, destroying your soil structure, destroying your soil health when you have another option? And then potentially something like chicory, which was showing that potential beneficial reduction in yield, which is a weird phrase to say, but that's how our industry is. I would say why not try it? I think there's a lot of perception that people are really afraid of the cover crops getting close to the fruiting zone. And I definitely mowed at the first site because it was a commercial vineyard, but I did not touch anything at the second site. I just let it grow, including those flowering stalks of chicory. And while maybe management-wise it wasn't pretty and offered a little few more difficulties, at the end of the day, though, that was about as minimal maintenance as I could, and it was still a pretty well-established system. So I say what I would say from growers is that it's unlikely you're going to be seeing dramatic effects. Why not try it and see what works for your system? Yeah. The questions are growers ready to change practices. The world of viticulture is notorious for its the slowest rate of adoption curve ever in terms of any agricultural field. We are based in history, right? Rules and how we grow things, how we make our wine. That's literally law on how you make your wine in Europe. Are growers ready to change? I think some are, and there's always early adopters who benefit from this kind of thing. There's definitely one grower in this region who plants buckwheat not only as an inter-row cover crop, but just everywhere, under vines, inner rows, everywhere. He's willing to try it. You'll always have those people who are. And I think it's stepping away a bit of that perception of that very nice-looking vineyard of having vegetation-free, and stepping more into maybe more of the ecosystem management perspective. At the end of the day, vineyards are also places where people host weddings and those sorts of things, and it's always hard for me to remove that when I'm at the end of the day asking a grower to do something a little different. For the chicory? Yeah. Good question. I think even though the resulting vine water status isn't that different because these are but these are vines that, while grown with some irrigation, I still think these roots are going incredibly deep, as we can see often in the Finger Lakes. And I think maybe while the whole vine water status isn't being that affected, they are having to rely on drawing water from those deeper roots than the upper surface of the roots where the cover crop is actively growing. And I think there might be some of that just physiological differences that are causing some of that. Similarly, it is known that pre-veraison water deficits will significantly impact fruit set. That's Mark Matthews' work. And I think maybe there were just some differences, but just so slight that statistically they weren't there. But I don't really have a great question or answer to that, unfortunately. But I think it has a lot to do with just the sensitivity of my measurements and maybe some physiological processes going on in the vine that I'm not able to detect. Sure. I established my cover crops by hand seeding and hand cultivating over them. It's not that glorious, but it was efficient. They sufficiently emerged every year. If this was to be adopted on a larger scale, we understand most growers are not going to be willing to walk over their acres and acres of vineyards with hand seeding. But there's interest and potential with a few of our collaborators in the extension side of things that have been coming up with different ideas of how to implement this on a vineyard level scale. But yeah, no, that's right now is just me, myself, and I of labor of hand seeding. Yeah. So the questions about the site to site differences, which there certainly was. Definitely, I think there were differences by the nature of the age of the vines. As I said, that first site was almost 20-year-old vines. And the second site here, these vines are younger, and I think it's just a little less fertile, a little less deep soil of a site. What is interesting to note, though, the one carryover cover crop of buckwheat pretty much had no significant effects at either site. So that's sort of my take-home message. There's going to be more and less vigorous sites here in the Finger Lakes. We have vineyards planted on the whole range of them here. So at the end of the day, I was trying to look at more take a step back from what I can say at this particular site and what could I say about the general trend of using undervine cover crops as a whole. But yeah, there certainly were site differences, and that's something to consider for any grower, if they were looking to change up their vineyard floor management, is how their site may differ. But both these sites were relatively fertile, and both sites were exhibiting excessive vegetative growth, so that's why they were selected. Any more questions? Well, thank you all for coming. Please help yourself to any food remaining, and I'm up here if you want any further questions.
This has been a production of Cornell University, on the web at cornell.edu.